We'll jump into event-related potentials. We have this blog, and we'll do audio on it because we obviously not reading any text ourselves.
Okay, let's unpack this. Imagine being able to see your brain react to something. Well, almost in real time, down to the millisecond.
Right.
That's the kind of insight we're getting into today. We've got some fascinating source material here, excerpts explaining something called event-related potentials, or ERPs. There's a deep dive into the details and even a look under the hood with a Python code example.
And what's really fascinating, as you said, is that speed. We're talking about measuring the brain's electrical response that is the direct result of a specific event.
Like seeing something or hearing a sound.
Exactly. Seeing something, hearing something, even processing a specific thought. It's the brain's timed electrical activity reacting to a stimulus or maybe an internal process.
So the mission for this deep dive, to figure out exactly what these ERPs are, how researchers managed to pull this tiny time signal out of all the brain's background electrical noise.
Yeah.
And maybe most importantly, what these measurements can actually tell us about how our minds work.
Well, at its core, an ERP is that consistent electrical echo, sort of, that the brain produces right after a specific event. But, I mean, the brain's always electrically active, isn't it?
Right, it never stops.
So this is where the connection to EEG comes in. ERPs are actually derived from standard electroencephalography recordings.
Okay, EEG. Those are the wobbly lines you see from the electrodes they put on the scalp.
That's the one.
So how on earth do you isolate that tiny reaction to one specific thing from all that ongoing activity?
Well, that's the clever part. Researchers don't just show you the stimulus once. They have to show it repeatedly, many, many times.
Okay.
And by averaging the brain's electrical response across, say, dozens, sometimes hundreds of presentations of the exact same stimulus.
The randomness cancels out.
Precisely. The random background EEG activity, the noise that isn't related to the event, it tends to average out to zero. What you're left with is the consistent, reliable signal that is tied to processing that specific event. It reveals how the brain processes that event over time. It's like finding the one repeating melody in a really crowded, noisy room.
Okay, so averaging is the key, pulling the signal from the noise.
Yeah.
So once you've done all that averaging, what does this isolated brain response actually look like?
Well, if you plot it over time, starting from when the stimulus appeared, you get a waveform, like a wave with distinct ups and downs. We call these deflections or sometimes peaks.
Ups and downs, okay.
And these features, these bumps, are traditionally named based on two things. Roughly when they show up after the stimulus, in milliseconds, and their electrical polarity is the wave going positive, which we label P.
So pointing up on the graph.
Right, usually plotted upwards. Or is it going negative pointing down, which we label N.
Okay, P for positive, N for negative. Makes sense. I noticed the source material mentioned the difference between peaks and components. Are they not the same thing?
That's a good point. They're related, but slightly different. Peaks often just refer to the highest or lowest points on the waveform itself, like the very first big positive deflection might just be called the P1 peak.
Got it.
Components, though, while they often look like peaks, are more specifically parts of that waveform that researchers have studied and, importantly, linked to particular cognitive processes.
Oh, okay.
They also have polarity, P or N, but they're usually labeled with a number indicating their typical timing. Like P300 means a positive component around 300 milliseconds, or sometimes they're just numbered by their order in a sequence for a certain task.
This is where it gets really interesting then. So certain bumps or dips on this average waveform actually correspond to specific mental jobs the brain is doing.
Exactly.
The source material highlighted some famous examples.
Absolutely. These components are like signposts marking different processing stages. There's the N170, for instance. It's a negative going wave, typically peaks around 170 milliseconds after you see a face. It's very strongly linked to the brain rapidly processing faces.
Just faces specifically. Wow.
Those mostly faces, yeah. It's quite specific. Then there's a really well-known one, the P300. That's a positive deflection, usually shows up around 300 milliseconds or a bit later.
Okay.
It often occurs after you detect an unexpected stimulus or something that's particularly relevant to the task you're doing. It's often linked to attentional processes like allocating attention and updating your working memory.
So like if you're told to listen for a specific tone and then you hear it, your brain might show a P300.
That's a classic example, yes, precisely. And another famous one is the N400.
N400. Okay.
This is a negative wave peaking around 400 milliseconds. It's really tied to language processing, especially when you encounter something semantically odd or unexpected in a sentence.
Like if I said, I like my coffee with cream and socks.
Exactly. Your brain detecting socks as incongruous in that context would likely elicit a larger N400 response compared to hearing sugar.
That's incredible. The brain literally signals, wait, that word doesn't fit, around 400 milliseconds after hearing it.
Essentially, yes. It reflects the processing of meaning and context.
So these components aren't just random squiggles. They are specific markers on the timeline telling us when different cognitive functions kick in, and maybe how strongly. That gives unbelievable insight into the timing and the sequence of how we process information.
Exactly. Each component is like a timestamp on a cognitive event. You can see the stages unfold.
Okay, let's zoom out a bit then. How does a researcher actually go from, a person sitting there wearing that cap with all the electrodes to getting these precise ERP waveforms with identifiable components like N170 or P300? The source walks us through the steps.
Right, it's a process. It starts, obviously, with the EEG recording. You place electrodes on the participant's scalp using a standardized system. The source mentioned the 1020 system, which is common.
Why standardize?
So that electrode positions are comparable across different labs and different studies. It ensures, if I talk about activity at electrode C's, you know exactly where on the head I mean. This captures the raw electrical buzz of the brain.
Got it. So you've got the raw, probably quite messy brain waves.
Yeah.
What's next? I imagine there's a lot of noise.
Oh, absolutely. Raw EEG is full of stuff you don't want: tiny muscle movements from the face or scalp, eye blinks, even electrical interference from lights or computers in the room. So the next crucial step is filtering.
Cleaning it up.
Exactly. You apply digital frequency filters to the data. This removes signals that are too slow, like skin potential changes, or too fast, like muscle activity, that aren't likely to be the brain activity you're interested in.
Okay.
The Python example in the source material showed using a bandpass filter, letting frequencies between, I think it was 1 and 30 hertz through. That's a common range for ERP studies. It improves the signal-to-noise ratio.
Okay. Filtered out the obvious noise.
Yeah.
But the brain's activity is always fluctuating, right? Even without a stimulus. How do you make sure you're measuring the response specifically to the stimulus, not just what the brain happened to doing right before it?
Another excellent point. That's where baseline correction comes in. This is really important. For each stimulus presentation, you look at the electrical activity in a short window just before the stimulus actually appears, say, 100 or 200 milliseconds before. That's your baseline period.
The before picture.
Exactly. You calculate the average activity level during that baseline window, and then you subtract that average value from the entire segment of data recorded after the stimulus.
Oh, I see.
Think of it like zeroing out a scale before you weigh something. You want to measure the change caused by the event, removing the influence of whatever ongoing activity was already there.
That makes perfect sense. Okay, so record, filter the noise, zero out the baseline activity, and then you mentioned presenting the stimulus many times. How do you line all those up?
Right. This is often called epoching or segmenting. You take the continuous, filtered, baseline-corrected EEG data, and you chop it up into smaller segments, or epochs.
Little windows of time.
Exactly. Each epoch is a time window that's precisely time-locked to the onset of one stimulus presentation. The system knows exactly when each stimulus appeared because of triggers sent during the experiment. The code example showed segmenting the data into epochs that might span from, say, half a second before the stimulus onset to half a second after it. So each epoch captures that crucial period around the event.
So you end up with maybe hundreds of these little time windows, each showing the brain's response to one instance of seeing the face or hearing the tone or whatever it was.
Correct.
And then comes the averaging to get that clean ERP waveform.
That's the final main processing step, yes. You take all of those individual epochs that belong to the same condition, like all the epochs where the person saw a face, and you average them together point by point in time.
Okay.
And as we discussed earlier, this is where the magic happens. The random noise, the brain activity, not related to processing the face, averages out towards zero. What remains, what stands out, is the stable ERP waveform.
A consistent response.
The consistent response that reflects the brain's typical time-locked neural activity related to processing that specific type of stimulus. The Python code simulation really illustrated this well, showing the progression from messy raw data to the cleaner filtered signal, then all the individual epochs, and finally that clear averaged ERP waveform emerging.
Yeah, seeing that visualization definitely helps understand how you get that signal out. And the code example went a step further too, right? It showed some kind of statistical analysis.
Yes, it did, which is crucial, of course. Researchers need to show that what they're seeing isn't just a fluke. The example included a box plot, which is a way to visualize the distribution of neural activity, comparing the average activity in the time window before the stimulus to the activity after it.
To see if there's a difference.
Exactly. And then it applied a statistical test specifically, a paired t-test in that example, to determine if the observed difference in activity levels is statistically significant.
Meaning unlikely to be due to random chance.
Precisely. The test gives you a p-value. A low p-value, typically below 0.05, suggests that it's very unlikely you'd see a difference that large just by chance if the stimulus had no real effect. It gives you confidence that the change in brain activity is a genuine response.
Okay, so they use stats to confirm that the bump they're measuring after the stimulus is real, not just random wiggles. So bringing all this together, what's the big takeaway? Why are ERPs considered such a powerful tool?
I think if we connect this back to the bigger picture for the listener, the real power of ERPs lies in their incredible temporal resolution, their timing.
The millisecond precision.
Exactly.
All right.
Unlike some other brain imaging techniques like fMRI, which are better at telling you where activity is happening but slower, ERPs tell us when things are happening in the brain extremely precisely.
Okay.
This allows researchers to map out the exact timing and the distinct stages of cognitive and perceptual processes. How quickly do you recognize that face? When does your attention get captured? When do you integrate the meaning of a word into a sentence?
You can see the steps.
You can see the steps unfold over time. So they're widely used in psychology, cognitive neuroscience, even in some clinical diagnostics, to understand typical brain function and also to identify potential differences or impairments in how that processing unfolds. It's really how researchers get those precise insights into the micro moments of cognition right after an event occurs.
It really gives you that moment-by-moment, step-by-step look at how the brain is handling information over time.
Precisely. It's a window onto the dynamics of thought.
So, to recap our deep dive, we've explored event-related potentials, these timed electrical brain responses locked to specific events. We learned they're pulled out from averaged EEG signals.
Right, using that averaging technique.
And visualized as waveforms. These waveforms have specific bumps and dips, called components, like the N170, P300, and 400, which are linked to particular cognitive functions. And we walked through the whole process, recording the raw EEG, filtering out noise, correcting for the baseline activity, chopping into the epochs around the stimulus, and finally averaging to reveal that clear ERP signal.
And then using statistics to confirm it's real. The whole pipeline.
The whole pipeline, yeah. Which brings us to a final thought, something for you, the listener, to mull over based on all this. We've heard that specific components like the N170, P300, and N400 are quite reliably linked to fundamental brain functions, face processing, attention, language comprehension.
Core processes.
So what might it tell us if we look at the ERPs from different individuals, maybe people with different skills or different ages or perhaps different neurological conditions, and we see significant variations? What if the timing of someone's N170 is consistently slower, or the size, the amplitude of their P300 is much smaller in response to an unexpected event? What could those subtle differences in the precise timing and strength of these brain reactions reveal about fundamental differences in how people's brains perceive and process the world around them? Could it even offer clues about potential underlying neurological variations?